Welcome to the Deep Dive. We're the show that helps you get straight to the core insights from the latest research. And today we're tackling something really significant, a medical frontier affecting millions, neurological disorders. Things like stroke, Alzheimer's, Parkinson's, MS, or maybe a spinal cord injury. These aren't just difficult conditions, they're actually the leading cause of disability and the second biggest cause of death globally. It's huge. And the fundamental problem, really, is that our neural tissues, the brain, the nerves, all that intricate wiring, they just have a very, very limited ability to heal themselves. Current treatments, they often just manage symptoms, maybe slow things down a bit, but they rarely fix the underlying damage. It's a huge gap in medicine. Okay, so let's dig into this because there's something really interesting happening, maybe even a revolution brewing that could potentially change things. Today's deep dive, it's all about 3D bioprinting for neural repair. It sounds like sci-fi, but it's emerging as a potentially transformative solution.
Exactly. And a lot of our discussion today is drawing from a really excellent, very recent perspective article. It's from 2025 by Mitchell Sinclair Glover, Zillian Yu, and Mirella Dettori, published in Advanced Materials. It really gives a fantastic overview of where the field is right now.
Right. And our mission for you today is basically to unpack how this, cutting edge tech actually works. What are the really big challenges it's facing? And crucially, what's the groundbreaking promise here for the future of medicine? Think of this as your fast track to understanding what might be next in healing the brain and nerves. It's moving incredibly fast, and you're about to get the inside view. So maybe let's start with the basics. What is neural tissue actually, and why is it so notoriously difficult to fix? Well, your nervous system, it's this vast, incredibly complex network. It integrates, controls, coordinates pretty much everything in your body. You've got the central nervous system, the CNS, that's the brain and spinal cord, and then the peripheral nervous system, or PNS, that's the huge network of nerves spreading out everywhere else. And down to the cellular level, it's mostly built from two main players: neurons, signal transmitters, zapping messages
Aren't
around.
SAS talkers?
Exactly. And then glial cells, which are kind of the support crew. They clean up, provide structure, help neurons function properly. The unsung heroes, really.
And that difference between the CNS and PNS, it's absolutely critical when we talk about repair. See, the peripheral nervous system, it actually does have some natural ability to repair itself, nerves in your arm, for instance. But the central nervous system, the brain, the spinal cord injuries there, like from a stroke or spinal cord damage, are largely non-regenerative. That's precisely why those injuries are often so devastating. The main highway, if you like, just can't rebuild itself once it's broken.
And for years, how did scientists even study this stuff? Mostly animal models or cells grown flat in a dish, right?
That's right. Those methods were fundamental. Definitely, we learned a lot about development, function, disease, but they have real limitations. There are ethical concerns with animal models, of course, and, well, animal systems aren't identical to human ones. And those 2D cultures, growing cells flat on plastic. It's simple, it's cheap, but it just doesn't capture the complex 3D world inside our bodies. It misses all those crucial interactions between cells and their surrounding environment, the matrix. That's key for how tissues actually work.
Okay, so that brings us to stem cells. That was a big leap, wasn't it? Human pluripotent stem cells, or HPSCs.
A massive leap forward, yes. These HPSCs are essentially biological blank slates. You can nudge them, coax them into becoming almost any cell type you need, including all sorts of neural cells, different neurons, glial cells from both the CNS and PNS. Using models derived from these HPSCs has really pushed our understanding of diseases like Alzheimer's or Friedreich's ataxia. It gives us a much more human-relevant way to study them compared to those flat cultures.
Right, and here's where it gets really interesting, I think. Moving from 2D into 3D with things called organoids. What are those exactly? Mini brains?
Sort of, yeah. Think of them as tiny, self-organizing clumps of these stem cell-derived neural cells. They offer a much better environment, more like what you'd find in vivo inside the body. The cells mature better, interact more naturally. For instance, with Alzheimer's models, researchers found these organoids replicated the disease features much better than the 2D cultures. So they're a much more accurate platform for testing drugs, for example.
But even these mini organs have drawbacks.
They do! Organoids are impressive, no doubt. But their big limitation is that they self-organize. It sounds good, but it means you don't have precise control. You get variability between batches, between labs. It's hard to standardize. You can't tell cell A to go here and cell B to go there. And that's the gap that 3D bioprinting aims to fill.
Okay, so how do we get that real 3D structure, that precise architecture we see in actual tissue, This is where the bioprinting comes in properly.
This is exactly where it comes in. Imagine like a very sophisticated 3D printer, but instead of plastic, it's precisely depositing living cells mixed with special biocompatible materials. We call these materials bioinks, and it builds the tissue structure layer by tiny layer.
It's like an inkjet printer for biology, but super precise.
Kind of, yeah.
Yeah.
And the key advantage over organoids, or any other method really, is that control.
That precision.
Unparalleled control. You can design the tissue architecture, you can place specific cell types exactly where you want them, along with other things like growth factors. This lets us create really intricate shapes, complex arrangements of different cells, trying to truly mimic the structure and hopefully the function of native tissues. It's a huge step towards recreating the body's own blueprints.
It still sounds a bit like science fiction, honestly.
Yeah.
But okay, making it actually work, especially for something as delicate and complex as neural tissue, that must be incredibly challenging. What are the big roadblocks?
Oh, the hurdles are definitely immense. First off, there's the sheer biological complexity. Neural tissue isn't just one thing. It's packed with the different types of neurons, different types of glial cells, all arranged in very specific ways. Think about the neocortex in your brain. It has 6 distinct layers, each with its own unique cell population and connections. Replicating that, placing all those cells in the right spots and the right numbers, that's a major challenge.
Wow, okay. 6 layers. Yeah, that's complex.
Yeah.
What else?
Another big one is a vascular and immune integration. Basically, any chunk of tissue bigger than a fraction of a millimeter needs a blood supply, needs oxygen, nutrients. And getting blood vessels to grow into a printed construct naturally is very slow, way too slow for larger tissues. The cells in the middle would just die.
They'd starve.
Exactly. And you also need the right immune cells integrated, like microglia in the brain, to clean up debris, manage inflammation, help the tissue integrate properly. It needs to live within the body, not just exist.
But researchers are finding ways around that, aren't they? I've read about printing tiny channels, almost like pre-made blood vessel networks.
Yes, that's a really active area innovation. Things like coaxial bioprinting, where you print a tube within a tube, or using sacrificial materials that you print and then wash away to leave channels. There are some amazing examples, like creating triple-layered vessels or conduits that mimic brain vasculature. It's absolutely vital for keeping larger constructs alive.
Okay, so complexity, getting blood supply.
Yeah.
What's next?
The third major challenge is biomaterial constraints. The materials you print with the bioinks are critical. Neural tissues have really unique physical properties. Your brain is incredibly soft, almost like jelly, but a peripheral nerve, much tougher, much stiffer. The bioink has to match the mechanical properties of the target tissue. If it's too stiff or too soft, the cells won't behave correctly. They won't function properly or integrate well.
So what are they using? What are these bioinks made of?
Mostly hydrogels. These are polymer networks that hold a lot of water, kind of mimicking the natural extracellular matrix, the stuff between cells in our tissues. There are natural ones like collagen or hyaluronic acid, and synthetic ones that offer more control over properties like stiffness or degradation rate. Often, the solution is hybrid bioinks, trying to combine the best of both. Plus, people are exploring adding conductive materials like graphene to help with electrical signaling and making sure the scaffold degrades controllably over time, letting the body's own tissue take over.
Right, it needs to disappear eventually. Okay, and one more big challenge you mentioned.
Yes, validating function. You can print a beautiful structure, but how do you know if it's actually working like real neural tissue? Is it firing signals? Are the networks connected? Traditional methods we use for cells in a dish, like calcium imaging or sticking tiny electrodes onto cells, patch clamping, they're really difficult to do effectively deep inside a dense, opaque 3D structure. You often can't see what's going on without damaging it.
So you need better ways to peek inside, essentially, non-invasively.
Exactly. That's why there's a huge push for things like integrated sensors, tiny microelectrode arrays, MEAs built into the structure, or stretchable bioelectronics or embedded optical sensors. These could let us monitor the electrical activity and the health of the tissue in real time, non-invasively. That's crucial for knowing if these constructs are truly functional before we even think about putting them in patients.
So yeah, these are definitely not trivial problems. Complexity, blood supply, materials, checking function. But it's fascinating how these hurdles are actually driving so much creativity, pushing engineers, biologists, material scientists to all work together. It really feels like a frontier.
Okay, so despite those really significant challenges, why is everyone so excited? What is the actual promise of 3D bioprinting for neural repair that makes it worth tackling all this?
Well, a huge advantage is creating those bespoke architectures and being able to functionalize the scaffolds. Bioprinting gives you that unparalleled control to build customized tissues, really trying to replicate the specific microenvironment that cells need. For example, researchers have built multi-layered human brain models with specific zones of neurons and astrocytes. They found these formed functional networks quite quickly, faster and more controllably than organoids. It opens the door to modeling really specific complex structures, like parts of the inner ear, the cochlea, or specific regions of the spinal cord with much greater accuracy.
So better models for research and testing. Definitely. And beyond that, bioprinting is great because it can be combined with complementary technologies. It's a very modular platform. You can integrate things like electrostimulation directly into the printing process or the culture system. This can help promote nerve growth, guide axons, help the cells mature, even mimic things like deep brain stimulation used clinically. You can also use bioreactors, maybe spinning bioreactors like those used for organoids, to improve nutrient flow and keep larger constructs healthy long term. Or integrate microfluidic devices for precise environmental control. It all works together to make better, more functional tissues.
Okay, let's connect this to the bigger picture then. What does this mean for actual patients? What are the potential real-world applications?
There are several key areas. First, as we touched on, is therapeutic testing. These bioprinted models are fantastic tools for studying diseases like Alzheimer's, Parkinson's, even brain cancers like glioblastoma in a more physiologically relevant way. And importantly, they enable high-throughput screening for new drugs or testing for neurotoxicity. You can test many compounds quickly and in a standardized way. This could massively speed up drug discovery and reduce the reliance on animal testing. There are already platforms being developed for this, like the Rastrum platform.
Faster drug discovery, fewer animal tests, that sounds good.
What else? Second, there's the idea of using cell-free bioprinted scaffolds to help the body heal itself, what we call endogenous repair. This is for injuries where there's still some potential for the existing tissue to recover, if given the right support. In the peripheral nervous system, think about large gaps in nerves, maybe after an accident. Bioprinted nerve guidance conduits, NGCs, could be a major advance over current methods. Instead of just a hollow tube, you can print conduits with specific internal structures, maybe channels to guide nerve growth, and load them with growth factors like NGF or GDNF. Preclinical studies in rats show these can work as well as traditional nerve grafts, which involve taking nerve from somewhere else in the body.
So guiding the body's own repair crew, essentially.
Exactly. And for the central nervous system spinal cord injury, traumatic brain injury, similar ideas apply. Scaffolds can provide physical guidance for any regenerating axons, help reduce scar tissue formation, which inhibits repair, and deliver therapeutic molecules right where they're needed. There's work, for instance, using collagen scaffolds for spinal cord injury that improved locomotor function in animals.
Okay, and the third major application area, that sounds like the most ambitious one.
It is. This is using cell-laden constructs for actual neural tissue replacement. This is for when the damage is too severe and you need to bring in new cells, new tissue. In the PNS, you could print scaffolds containing Schwann cells, the support cells that help nerves regenerate and myelinate to bridge really large gaps. Or think about innervating other engineered tissues. If you make artificial skin, it needs sensory nerves to be truly functional for wound healing. Engineered muscle needs connections to nerves to contract. Organs like the bladder need autonomic nerve control. Bioprinting could provide that neural component.
Wow, so printing nerves into other artificial tissues.
Yes, and in the CNS, this is where the potential is really dramatic.
Right.
Replacing damaged circuits after spinal cord injury, for example. There was a landmark study a few years back where researchers bioprinted scaffolds loaded with neural stem cells and implanted them into completely severed spinal cords in rodents. They showed it supported axon regeneration across the gap and actually restored some motor function. And they even suggested it could be scaled up for human injuries. That was really groundbreaking.
Incredible.
And you can imagine this for neurodegenerative diseases too. Like for Parkinson's, printing constructs containing dopamine-producing neurons to replace the ones that are lost. Or for Huntington's, trying to restore specific circuits and potentially using patient-specific stem cells to avoid immune rejection.
This all sounds incredibly promising, almost revolutionary. But this raises an important question. Beyond the amazing science, what else needs to fall into place for this to actually reach patients in the clinic? It can't just be about the tech, right?
Absolutely not. There are significant regulatory and ethical considerations to navigate. There are ongoing ethical discussions about the source of cells, embryonic stem cells versus induced pluripotent stem cells derived from a patient's own skin, for instance. As these models, like brain organoids, get more complex, there are even philosophical debates about potential consciousness, though we're very far from that. And safety is paramount. You have to rigorously test for any risk of immune rejection or the potential for the implanted cells to form tumors.
And the rules, the regulations.
That's a big one. The current regulatory pathways are mostly designed for traditional drugs or simpler medical devices. These complex, living bioprinted constructs don't fit neatly into existing boxes. So new frameworks are needed to evaluate their safety and efficacy. It's a challenge regulators are grappling with worldwide. But progress is being made. You mentioned there are already, 11 clinical trials involving bioprinting in some form? That's right, over a dozen trials are underway globally, testing various bioprinted applications, which shows the field is moving towards clinical reality, albeit carefully.
Okay, so let's try and wrap this up. 3D bioprinting. It really does seem like a genuinely transformative tool. It gives scientists this incredible, unprecedented control to create neural tissue, and that opens up amazing possibilities for modeling diseases better, finding drugs faster, and developing completely new regenerative therapies.
Exactly. And yes, huge challenges definitely remain. Replicating the full complexity of the human brain, that's still, a long way off. But these challenges are precisely what's driving this incredible surge of innovation in materials, in stem cell biology, in the engineering of biofabrication. It's pushing the absolute boundaries of what we thought was possible.
So the future here probably isn't some single magic bullet, a one-size-fits-all fix for neurological problems. Instead, what we're looking at is the possibility of highly tailored, personalized treatments driven by this amazing technology. Just think for a moment what that could mean for someone you might know who's living with a neurological condition, or, frankly, for the future of your own health down the line. It really isn't just about printing tissue, is it? It feels more like it's about printing possibilities for a healthier future.